Good afternoon, everyone. It's great to be here. It's the third LHON patient day that we're holding this time, and it's fantastic. We had the first one at Moorfield 3 years ago now, or more, and then the last one was in Cambridge 18 months ago, so it's great seeing new and old faces in the audience. So in the next, I guess, half an hour, what I want to do is just cover some of the latest update on treatment options for Leber's optic neuropathy. I know there are people here in the audience who have not been to previous patient days, so I thought I would just give a brief overview first and then give you some of the newer results, including some of the early results coming from the gene therapy work being done by Gensight. So in terms of my disclosures, I don't own any stocks or shares in the various companies that I'm going to mention. I'm quite closely involved with the Gensight gene therapy trial, and I'm also working very closely with Santhera and more recently with Stealth Biotherapeutics. So it's been a very long journey. So this condition that we're talking about today is named after Theodor Leber. He was a German ophthalmology, worked in Heidelberg University. And basically in 1871, he wrote this monograph of 4 families with a very classical a presentation of Leber's optic neuropathy. So even then, without genetics, without the fancy technology that we have now, he was able to pinpoint some of the salient features of this condition. First of all, that it is men that tend to be affected. Secondly, that it tends to affect young people. And then thirdly, that this has a reasonably a rapid progression and quite a poor visual prognosis. And then we need to fast track many years down the line. And it's in 1988 that Doug Wallace and his group, and also Ivan Ikoskilainen in Finland, came up with this seminal paper in Science describing for the first time a pathogenic point mutation within the mitochondrial genome causing Leber's optic neuropathy. And this changed the game completely because it really ushered in the era of mitochondrial medicine. I don't have to say this to the audience, but this is a condition which really has a major life-changing effect on both the patient and the wider family. There's very rapidly progressive loss of vision in both eyes, and we know from this study that we did 10 years ago now with my good friend and colleague Patrick Chinnery, and also with a student working with us at the time, Matt Kirkman, that this has a big impact on someone's quality of life. And if you rank all the other ophthalmological disorders, LHON is the worst in terms of the visual impact. So this has been the focus of a lot of research, both here in this country and also in other parts of the world. It's really about how we can rescue the optic nerve in this condition. Because what is quite intriguing about Leber's optic neuropathy is that this pathogenic mutation in your mitochondrial genome is present in every cell of the human body. But for some reason, the cells that constitute the optic nerve, these retinal ganglion cells, are the ones that get hit very early and very hard. And fortunately, most of the time, these are the only cells that get affected. So this is a bit of an artificial divide, but what I've done for this presentation is split into 4 parts. One is the loose labels of mitochondrial antioxidants. The second part is how we can activate the mitochondrial machinery to try and improve the function and therefore hopefully rescue these cells. The third, which is very topical, very sexy and very promising, is gene therapy. And the fourth, very briefly, is just to discuss a little bit about how difficult and challenging it is to do research for rare diseases. And part of the reason why things are slower in the field of rare disease is because resources can be limited, and also the process of recruiting patients can be much more challenging. So let us start with the first big umbrella topic, which is mitochondrial antioxidants. So the main one, and the one that you will all have heard about, is this compound called idebenone, which is being marketed by Santhera. And this is a very interesting compound because it is a synthetic molecule, and what it does fundamentally, it helps to push electrons along the mitochondrial respiratory chain, which is essentially the little machines within your mitochondria that produces energy, right? So if you're able to push these electrons, you're able to improve the function of these mitochondria, and the rationale is that you will improve the amount of energy being produced. And this is correct because when we look at the data produced in cells, like we take a little skin biopsy from patients that have volunteered. We grow this in the lab, and if we supplement the culture media with idebenone, there's good evidence that you improve the amount of energy being produced, and you also decrease the levels of these toxic reactive oxygen species. So in collaboration with Santhera, who had the foresight of doing a randomized control trial, back in 2007, roughly speaking, we started recruiting patients into the RHODOS trial. And the study was published in 2011, already 8 years down the line. And the bottom line is that the primary outcome measure was negative. And we can talk a bit more about why that was the case. But fundamentally, there was consistent evidence of visual benefit for a subgroup of patients that were treated. And there was also this other paper coming out from the group of Valerio Carelli and Piero Barboni in Bologna, Italy, where they look at a retrospective case series. And again, the punchline is that idebenone had an effect on the visual outcome. And if you treated someone early enough, you had a much greater likelihood of improving visual function, which makes sense because if we're talking here about a disease that affects neuronal cells, time is of the essence, and the earlier you treat, the more likely you're going to recover some of these cells further down the line. What was also quite clear from these studies is you're not able to prevent the second eye from becoming affected. In roughly around 25% of cases, both eyes get affected very quickly, so it's not possible to say whether it's the right eye or the left eye. But in other cases, you have one eye which gets affected, and roughly around 3 to 6 months later, the other eye has gone. It would have been nice to have a drug that can prevent that, but certainly idebenone can't do that. And also, the consensus among the experts in the field is that there is no evidence to use idebenone in the context of someone who carries the mutation, but is an asymptomatic carrier. Now, after the RHODOS study was published, Santhera had an expanded access program. What it means is that across the world, in various countries, Raxone was being provided to patients, and over the years, data was being collected as to whether or not there was any change in their vision, side effects, etc. And this is really, to me, one of the main findings from this expanded access program. First of all, is that it does concur with the data that was generated from the RHODOS trial. So if you look at a group of patients with LHON who do not take idebenone, there's roughly a 10% chance of them recovering 10 letters on the vision chart. But if you are on idebenone, your chance is not 10%, it's more like 30%, right? Which is, still, not that great, because this means there's a percentage of patients who do not have any benefit from idebenone, but certainly it's much better than without treatment. So based on this cumulative evidence, RHODOS, the Expanded Access Program, the European Medicines Agency, which controls all the drug regulation in this country, they used to be based in London, but now they've relocated to Amsterdam. In June 2015, they decided to grant a marketing authorization to Santhera under exceptional circumstances, recognizing the fact that all the evidence was not there, but also being aware that this was a rapidly progressive form of blindness and there were no other treatment out there. And because of the uncertainty about whether or not idebenone should be used and when it should be used and how long it should be used for, a group of people with a big interest in inherited optic neuropathies, including Leber's optic neuropathy, got together in Milan in March 2016. And basically it was to really to thrash out the evidence and what our views were, both evidence and eminence-based, and to come up with an international consensus guideline, which was published about 2 years ago. And the Journal of Neuro-Ophthalmology will provide you with a copy if you go to their website. The most important take-home message from the consensus guideline is that there was evidence to use idebenone in the context of Leber's optic neuropathy, and the treatment should be started as soon as possible. And the main evidence is really if you have disease duration of less than 1 year. This is the group that should be targeted in terms of a potential benefit from using idebenone. Now, it's been a lot of work. If you think back that the trial started roughly around 2007, we're now 12 years down the line. If you think about all the preclinical work that was done before, it's a rather sobering fact that up to now, at least in England, we still are not able to prescribe idebenone, i.e. Raxone. And the reason for this is that both in this country and elsewhere, there can be a big gap between getting the evidence for efficacy of a particular drug or intervention and actually getting that to be reimbursed by your local national health authority. And I was just having a very interesting discussion just now at lunchtime with Marcel Vitruba, Russell, and with Kamlesh, Seth. And part of the process is economic, because we don't have a huge unlimited pot of money, and also partly because for rare disease we're still pretty much in uncharted territory to some extent. So in the context of Raxone, it's not going down the NICE route, right? So NICE says, well, that's not going to be within our remit. So the evidence for Raxone and whether or not it will get reimbursed in this country is going down the NHS England route. So both myself, Russell, some of the clinicians are part of this policy working group where we spend time reviewing the evidence and writing our recommendation. This then goes to various other committees within NHS England, and the process is still ongoing now. As to whether or not we'll get a decision by the end of the year, I really don't know, but I suspect there might be further delays. But the reason for me putting this forward is people like myself spend a lot of time thinking about potential research projects about how we can test X or Y drug. But what has become much more, part of my academic life is really also how we navigate the process of getting new drugs onto the market, which is challenging, and I'm learning every time I speak with someone who knows more about this rather convoluted process to some extent. Again, we were involved, the UK Election Society was quite closely involved with the Scottish Medicines Consortium, where things happen a bit faster, I suspect, because Scotland gets a bit more money per capita than England. But in Scotland, Raxone is available for NHS use. The criteria, we can argue about this, but the criteria is also not that helpful to some extent, because it can only be provided to patients who do not meet the UK criteria for severely sight impairment, which is basically a small group, because a lot of patients who do present with LHON have already been registered as being severely sight impaired, which is the old term for blind registration. But at least the process went through and it's now available for people north of the border. As part of the overall picture for idebnone and Raxone, there is also a study that has been running for the past 2 to 3 years called the LHON study, which was part of the requirement of the EMA, the European Medicines Agency, insisting that Santhera had to provide more information about the efficacy of idebnone, and also getting more data on safety profile, which we know is not an issue because idebnone is a very safe drug. So LHON has been ongoing in various countries, including here in the UK. Recruitment is nearly finished now. And essentially, the patients that were being recruited had disease duration of up to 5 years, and they were provided with treatment with Raxone for a period of 2 years. And I think this is going to be a very important study because the numbers recruited are significant. I don't know exactly how many have been recruited so far, but it's probably around the 150 mark. And if we have data on 150 patients for 2 years, this is going to be a very powerful way of analyzing whether or not there is a treatment effect, which is much more than what we would expect from the natural history. I don't really know the timeline for the analysis of the data, but I suspect we should be getting something by 2020. There are a few other drugs out there. If you read the published literature about drugs that could potentially improve the visual function in Leber's optic neuropathy, there's a drug called EPI 743 that was originally investigated by this company called Edison. Edison now no longer exists. It's been rebranded as Bioelectron Technology Corporation. I'm not sure what's happening in terms of their plans for Leber's optic neuropathy, so I'm not going to say any more about this. But there is also another biotech company called Stealth Biotherapeutics. They're based in Boston. They have a drug known as MTP131 or elamipretide, and they are heavily involved at the moment in mitochondrial diseases, including Leber's optic neuropathy. So as you've heard already before, the key issue with Leber's optic neuropathy is that there is a dysfunction of these mitochondria, the so-called batteries of your cells. And within these batteries, you have these little pockets, these little units that produce energy, right? But they don't work in isolation because they work very closely together. It's a like very intricate chain. So if one part of the chain is defective, everything gets messed up. So what I've been told about amiprotide is that this molecule allows the chain to be stabilized. So even if you have a mutation that causes Leber's optic neuropathy, if you're able to stabilize this mitochondrial chain, you're able to optimize the production of energy, and you're able to reduce the levels of these toxic reactive oxygen species. And by doing that, hopefully what you can do is you can treat and block the primary causes of the pathogenic mutation, prevent these cells from being lost. So certainly, I think this is an interesting compound. It's being used in various mitochondrial diseases at the moment, including mitochondrial myopathy and, of course, Leber's optic neuropathy. Time will tell whether or not it will be a game changer, but certainly this is an area where you'll probably be hearing more in the next 1 to 2 years. In terms of using this drug amiprotide in the context of Leber's optic neuropathy, a study has recently been completed in Los Angeles, led by Professor Alfredo Seidun. It was primarily a safety study where they were using a topical formulation of the drug. And the topical formulation is quite attractive in many ways because you put the drops in your eyes, no needles involved, etc. But the drug can also be administered subcutaneously. So time will tell what is the best way of giving the drug, whether it's by giving drops or by giving injection in your arm or in your leg. So the data is being analyzed at the moment, but certainly the hope is that the data generated from this early phase will lead on to a much bigger study involving various countries, including the UK, to test in a larger group of patients whether or not this drug has a future in the context of treatment for Leber's optic neuropathy. So this is the first part. I'm just going to say something very briefly now about a topic which some groups are working on, is how can we activate the mitochondrial machinery in a much more efficient manner. So we've already heard about the fact that mitochondrial disease can be quite heterogeneous. In the context of Leber's optic neuropathy, one of the big mystery is the fact that not everyone who carries a mutation will lose vision. So you have this percentage out there, roughly around 50% for a man, roughly around 10% for a woman, but this is very much an overall statistical analysis, because there's a very big variation, as you will know yourself, based on your own personal experience. You will have families, for example, on the left here, where there's only one affected family member. Whereas you have other families, like this big family from the northeast of England, where almost everyone will experience loss of vision at a very young age. So there's a very big variability in terms of the so-called penetrance of the disease. So there's also a lot of genetic work being done at the moment. That's why when you come to clinic, sometimes you will be asked for a blood sample, because we're comparing the genetic profile from affected and unaffected carriers to see whether or not there's a way of better predicting your risk of losing vision, because this could be very powerful in the future. Because let us say there is a treatment that works amazingly well and you're able to identify someone who's very high risk, then you don't have to wait for someone to lose vision before you can intervene. And I think this is where we should be heading in the future. And I don't think this is science fiction. I think the way that the research has accelerated the past 10 years, we could very much reach a time in the future where we can predict with much more accuracy what is your risk of losing vision. Is it low? Is it high? Is it intermediate? And the reason for mentioning this is the fundamental difference to some extent between men and women is our hormonal profile, right? There's no doubt about this, right? So could your hormonal profile have an influence on whether or not you're going to lose vision in the context of your genetic background, in the context of environmental exposure? So this work has been done primarily by the groups in Italy, the group of Valerio Carelli in Bologna, and also the group of Cola Giordano from Rome. They've done some really interesting work where they've been using, for example, skin cells collected from patients with various mutations, growing these skin cells again in the lab under different environments, with or without hormones, and using different types of hormones. And the data is pretty robust in the sense that certain combination of estrogen hormones can protect these cells and can prevent the cells from dying. So if you extrapolate that to the human system, this could explain why women are much less likely to lose vision, because you have much more estrogens floating around. The obvious corollary is whether or not, if estrogens is protective, whether or not a woman who is a carrier of a mutation, whether they are at increased risk of losing vision in the perimenopausal period. It is speculative, and that's something that will probably require quite a large epidemiological study to be able to tease out, but potentially there might be a second peak for female carriers. But putting this aside, which is purely speculative, I think there is a lot of potential identifying these estrogen-like molecules. The reason why I say estrogen-like, because obviously you can't give estrogen to a man because they're going to develop various side effects, including feminization. But if you're able to pinpoint the part of the molecule which has the beneficial effect, then pharmacologists are very adept at modifying these molecules to get the benefits without the side effects. But having said that, this is something which will take a lot of time. Most of these new drugs being worked on won't resurface in the clinical arena in the next 10 to 15 years, because that's the length of time you need from the preclinical work to the clinical trials to eventually getting it approved for use. So it's a very long game, really. So let's move on to the third part, which is about gene therapy. So I've been very heavily involved with gene therapy for the past 5 years, because it's been a real game changer in the field of ocular genetics for very obvious reason. Because if you think about it, right, if you compare the brain with the eye is easily accessible. We can inject things in the eye very easily. We have very good tools for measuring vision and for measuring the structure of the eye. So it's been a very good model for checking whether or not gene therapy can help in these inherited diseases. And you've heard in the news that last year the first gene therapy for an eye condition called Leber's congenital amaurosis, not to be confused, because very often there will be some confusion. I get referrals for patients who have Leber's congenital amaurosis, but it's a different genetic condition. But the bottom line is that they've used a gene therapy paradigm, and they've been able to show that this can improve visual function, or at least stabilize visual function. And last year FDA has approved Luxturna for patients with RP65 Leber's congenital amaurosis. The difficulty for Leber's optic neuropathy is that the delivery of the gene construct is not very straightforward, because, as Marcella mentioned already, there are two membranes for your mitochondria, an outer membrane and a much more convoluted inner membrane. So there's this physical barrier. But to bypass this barrier, some people have decided they're going to use a technique called allotopic expression. The name itself is not really that relevant, but what it means is that if you can't push something directly into the mitochondria, what you can do is you can insert the replacement gene into your nuclear genome, and you produce the protein, and then the protein has a little signaling sequence that says you have to go to the mitochondria, right? So you bypass the barrier, and it's the protein which gets into the mitochondria. So this has been worked on, and there's been a lot of work done on the safety of this strategy. The bottom line is that Gensight, which is a company based in Paris, has heavily invested into clinical trials to test whether or not this gene therapy vector GS10 could be the next step forward in terms of improving vision for patients with Leber's optic neuropathy. And I think the preclinical data is pretty robust. They've done work on rat models, on cells, and they've done toxicity studies in primates as well, which show that there is no major adverse event. So having had all the data, then they moved on into the human phase of their development program. So you might have heard about 2 gene therapy trials that have been ongoing for the past few years. The first one is called Rescue, and the second one is called Reverse. So it's using a viral vector, right? But it's not a viral vector that's going to damage the eye. The virus has been completely neutered, so it can't cause an infection. But the reason why we use a viral vector, because the viruses are very good at penetrating cells. Well, it's evolution. The reason why a virus causes disease is because they can punch a hole in the cell and they can integrate into the the cell structure. So the rescue study is for patients with LHON, with the 11.778 mutation. It's only the 11.778 mutation, and it's for patients who lost vision from 0 to 6 months, so an early phase of the disease. Reverse is for patients who have visual loss between 6 and 12 months, so a bit later on, but still in a window where we feel that there is all to gain for. And what the study involved was, we all have two eyes, except if we've had an injury in one eye, but the principle of that study is one eye was randomized to be injected with the active viral compound, whereas the other eye was just touched with a little cannula, and it's the sham eye, right? So one eye was being treated, one eye was not treated, which we call sham in the technical jargon. And that's where you see the difference really about doing clinical trials now compared with 12 years ago when we did the Rodin study, where it was much more difficult to recruit patients. For the Gensight trials for rescue and reverse, the recruitment process really was very efficient, right? The reverse study was completed in terms of recruitment in February 2017, whereas the rescue study was completed slightly later in July 2017. The reason why rescue took a bit longer is because it was for patients with disease duration less than 6 months, and therefore we had to wait for people to present, a new patient to present to clinic to be able to recruit them. So in terms of the outcome measure, because every time you do a study, you must have a way of detecting a change, either worsening or improvement. So we must have robust, objective, standardized ways of checking whether or not the drug is working or not. And in ophthalmology, to some extent, we're quite fortunate. It's not ideal. There's still a lot of work that needs to be done, but there are two main things we can do, right? The first thing, which is much more relevant, to all of us, is does it improve how much you see, right? And for that, there are two main ways we can check vision. Either you use the usual standard chart that you have at your opticians, which is looking at high contrast vision, or you can also measure low contrast vision. On the other side, besides vision, you can also measure and analyze the structure of the eye, right? And if all people in the audience who have been to eye departments have had it probably at some point in the past, where they scan the back of the eye, you see all these funny lines, all these sparkling little dots, and what it does, it scans the back of the eye very quickly and at very high resolution, and is able to measure the thickness of various parts of the eye, both the optic nerve and also the retina. And that's very helpful because it gives an indication of whether or not you're able to preserve the function of the eye, the structure of the eye. Sorry. So what about the data coming out from the reverse study, which is what I'm going to tell you about next, right? So for those of you who are able to see the slides here, I'm showing a graph which plots how the vision is changing in both the treated eye, which is getting the viral injection, and also the sham eye, which is not getting anything, right? So if you look at 72 weeks after the treatment was given, the sham eyes without treatment improved by 12 letters, whereas the viral treated eyes improved by 15 letters. So it was quite unexpected that both eyes had an improvement, and I'm going to talk a bit more about this shortly. If you look at the data in the other way, in terms of how many eyes are achieving vision of 660 or better, 660 is essentially the top letter on the vision chart. So when you go to your optician, if you're able to make a top letter on the chart, this is 660. And the reason why we took this benchmark is it's unusual for patients with Leber's optic neuropathy to achieve vision better than 660 because it's quite an aggressive disease in terms of loss of vision. So if you look at these two groups again, if you were given a treatment, you had a 30% chance of preserving vision of 660 or better. Whereas if you were in the sham eyes, this was 16%, right? So 30% against 16%. And if you do all the fancy statistics, this is highly significant. So you are much more likely to achieve vision of 660 or better if you were treated with the viral injection. Now, if you look at the other metrics, there were a lot of outcome measures, and I won't be able to tell you all about this. I'm just picking what I feel are the more relevant ones. Now, if we look at the structure of the eye, what was quite interesting is that the eye that was being treated with the viral injection did much better. So you had a preservation of the retina and the optic nerve, which again suggests that something is happening here. And we also did some patient-reported outcome measures, and based on the aggregated data, if you, the patients that were recruited into the reverse study reported an improvement in terms of their visual function, which again is supportive of a treatment effect. Having said that, this is only the preliminary data, because the full data set for reverse will only be available in the third quarter of the year, and the full data set for rescue, which is 0 to 6 months, will only be available towards the tail end of 2019, and the results will be presented at the American Academy of Ophthalmology in San Francisco. Now, the first time I saw these results, I was like, what's happening here? Because we're only treating one eye. How come you're seeing an effect in the other eye, right? So could this be essentially noise and or a placebo effect, i.e. the treatment is not working? Yes, that's an option. The second option, which I think goes back to the fact that we're still learning a lot about the optic neuropathy, is whether, even as experts, we actually know what's the natural history of Leber's optic neuropathy. What by natural history we mean that if someone has Leber's optic neuropathy, loses vision, and then we follow their progression over time, not just for one patient, but for a big group of patients with LHON, how does the disease evolve? What's the chance of spontaneous visual recovery, and what's the likelihood of someone, for example, achieving vision of 660 or better? So if you look at the published data, right, there's been quite a few studies published over the years about what is the visual outcome in patients with the 11.778 mutation. The bottom line is that if you have the 11.778 mutation, the visual prognosis is not very good, because the chance of spontaneous recovery is probably hovering around the 5 to 10% mark. But the problem with these retrospective studies is people have used different criteria for defining improvement. What is improvement? What is actually really an improvement that will improve someone's functional quality of life? The best study out there is that study by Byron Nam and John Guy from Bascom Palmer, where as part of their own gene therapy program, they looked at 44 patients, all of them having the 11.778 mutation, and these patients were followed up, roughly speaking, every 6 months for a period of 36 months. So pretty good, really. It's a 3-year prospective study. So if you look at their data, they define improvement as an improvement of 15 letters, right? And the reason why they use 15 letters, because 15 letters has a bit of a magical feel when it comes to the FDA, which is the American version of the European Medicines Agency. So if a treatment improves vision by 15 letters, you can punch your fist in the air. It is working, and it's very likely that the FDA will give its blessing in terms of approving the drug. And 15 letters is about 3 lines on the vision chart. So if you look at the study by Byron Nam and colleagues, 13 out of 88 eyes, i.e. 15%, improve by 15 letters or more, which is not that great, really, right? And that's why Leber's optic neuropathy carries such a difficult challenge when it comes to visual rehabilitation. And roughly speaking, the average time for recovery was much later than I thought it would be, but it's around 28 months. So essentially, it's not always a conventional thing that all the improvement will happen within 12 to 18 months. You can get much later recovery of vision. So if you remember that number, so it's 15% for the prospective study from Bascom Palmer. But when you look at the reverse study, the percentage of eyes that were treated with GS10 that achieved 15 letters or more was around 41%. And this compares with the sham eyes, which was roughly around 35%. So based on this, the difference is quite marked, because it's 15% versus 41%. The next step is, could it be that you're injecting one eye and somehow you're getting some of the viral vector in the other eye. Well, the only way to do that is to do an experiment. So what the company is doing, it's investing quite heavily actually, because these studies are not cheap. They are doing some primate work. Whatever your views might be on animal experimentation, this is a accepted protocol for doing these studies in the scientific field. So they are injecting one eye, and then they will see eventually whether or not there is evidence of the vector having tracked somehow to the other eye. I can't give you the answer because the data is not yet available. The other possible explanation why you might be getting a bilateral improvement is there's also the visual cortex. So you have the eye, which is the camera, you have the optic nerve, which channels all the information from the eye to the brain, but you also have the visual cortex and all the association areas. So it could very well be that there is a reorganization of the visual cortex which allows the other eye to improve despite the injection having been done in one eye. So it's all hand waving, it's all speculative, but I'm just giving you the latest thinking process about this. Now, as mentioned already, the full data set for reverse will become available in the third quarter, and when this happens, an important thing will happen. Up to now, I don't know the code for the patient that have been recruited, so I'm completely blinded to the treatment, right? And to which eye has received the active treatment. But once the study is completely finished, the randomization code will be broken, and therefore every single patient that took part in the study will be able to be tracked individually. So it could very well be that we might see that there is a group of patients who do pretty well and others that don't do very well at all, really. In terms of the rescue study, again, this is just the top line data because we're still churning through the data set, and the full data set won't be available until the end of the year. And just to remind you, rescue is an earlier study from 0 to 6 months. And if you look at the rescue data, right, at week 48, in the treated eye, vision improved by 13 letters, compared with roughly around 12 letters for the sham eye. And at week 72, this had improved roughly around 21 letters. And 21 letters is a pretty, big improvement in vision. And the final thing which is ongoing now is the reflex study, which is the same, strategy using a viral vector. But this time, instead of only one eye being injected, both eyes are being injected. And this bilateral study has been recruiting now for the past 18 months, and we expect that recruitment will be finishing by next month. So again, the process has been pretty quick, and roughly around 90 patients will have received treatment with the GS10 vector. And there are 12 centers involved worldwide, and as I mentioned already, we will stop recruitment very soon. Now, it's not just Gensight out there. There's also the group of John Guy at Bascom Palmer who's doing work on mitochondrial gene therapy. We had the chance to discuss this recently, about 2 weeks ago, and he's also getting some promising data. And there's also the group of Professor Li Bin, who I actually first met for the first time last year at a meeting in Taipei. A very nice chap who's, I've not been to his lab, but he's actually heavily investing into research into LHON gene therapy, and you might be interested to hear that he's also working on gene therapy vectors for the 3460 and 1444 mutations. So that's something to follow for the future. Now, we can discuss a bit more about the results of the gene therapy program, but I think there are some promising data out there. We need to wait for the full data set to come out, and then we can sit down and basically digest the full data set in all its complexity. I think what has become quite clear, based on the translational work that we've been doing for the past 10 years or so, is that finding a cure is not going to be straightforward when it comes to Leber's optic neuropathy. Fundamentally, part of my worry is that once the disease process has started, whether or not we've already lost a lot of grounds here, and whether we're able to inflect the disease process sufficiently to make a marked difference in terms of someone's central vision. I think the data so far suggests that we can, but this is my own personal worry is, how quickly do we need to intervene to be able to have a real impact on someone's visual function. What is also quite clear, and that's something that we've discussed already, is, sometimes we are in our academic ivory towers, we're doing our research, and we're pontificating about X, Y, and Z, but, if you were the one doing the studying, having to finance this in the real world, it's a really big risk and a big ask, because you're spending 10 to 15 years in terms of drug development, clinical trials, investing hundreds of millions or billions of pounds depending on your patient group, and at the end you don't have any a guarantee that your drug will work. And even if it does work, whether or not your local national health organization will fund it based on cost. And also I think the one thing which we really need to get better at is what is causing the actual loss of these cells within the optic nerve, which is causing loss of vision. We've talked about loss of energy, we've talked about reactive oxygen species, but these are pretty generic end pathways, and we need to know much more about the complex process within the cells that lead to the cells dying in the first place. And there are various ways to do that. People have been using various models, mouse models, and now people are also using stem cells, but that's a topic for another discussion. And clearly, I think to be able to do these clinical trials, we need networks. Now we're quite fortunate that we have various societies across Europe. There's social media which is helping. You've also heard very briefly about the European Reference Network from Russell earlier. And I think this has a potential of really helping in terms of a pan-European registry of patients with Leber's optic neuropathy, and also how we can improve access to all these new clinical trials. It's still very early days. I think there's a lot of challenge out there, but I think we need to strive towards something which goes beyond the UK border, Brexit or no Brexit. So, and there's also the European Neuro-Ophthalmology Society, UNOS, which I'm heavily invested with, and we will soon have a meeting in Porto. There are a few people in the audience that will be taking part of this. And this is a picture taken at the previous UNOS meeting 2 years ago in Budapest, where it was very nice because we managed to get one session dedicated to patient networks. We got people from Sweden, we got people, Pablo from the Spanish Society, and we'll have another session again in Porto in June. I think fundamentally, unfortunately, I would like to think that I'm reasonably efficient, but even with a limited amount of sleep, I don't think I can do everything myself. And it's great now that we have people like Russell, who's very heavily involved with the various organizations like ERNI or Eurodis, and we also need you to get involved to be advocates and to basically challenge the stakeholders that LHON might be a rare disease, but it's a disease which has a major impact on someone's quality of life, and therefore we need more funding. And also fundamentally we need more funding in eye research, because I work very closely with neurologists. I have very good friends who work in rheumatology or in renal medicine. For something as crucial as our vision, we're very much underfunded, not just for rare disease, not just for LHON, but generally speaking for eye research, we're underfunded by the major funding bodies. And I think part of the problem is, we've not really been good at promoting what we can achieve in terms of all these major discoveries and these major seminal clinical trials. And it is something that we need to get much better at. And also this will help when the survey closes and we have all the data and can present that hopefully to members of parliament or the House of Lords or whoever is a prime minister when it gets published. That's on the topic really, right? So if you feel like coming to Porto, right, it is going to be in June. It's going to be very nice. Hopefully I don't drink myself, but I'm sure some people there will enjoy the good wine and the port wine. And this is my final slide really, that it is challenging, it's difficult doing research in rare disease, but more than ever I'm very optimistic. I started working in Leber's optic neuropathy about time flies. I won't say how long it has been, but it's been more than 15 years. But it's really changed, I think, from the pure genetics, from the pure how is this cell behaving in the lab in terms of X, Y, and Z. And it's really exciting now where you go to meetings, you speak with people who are thinking very hard about trying new drug molecules in the context of Leber's optic neuropathy, because it is a very elegant model of a disease affecting the optic nerve which happens very quickly, and therefore if a drug works, this could be applied to other optic nerve diseases like glaucoma, for example. So I think there's a lot of interest from the pharmaceutical industry that we can capitalize on. Thank you very much. And it's a very broad topic, and it's covered a lot of ground. So I'll be more than happy to take questions to try and tease out areas that were perhaps not as clear, and that you might want to get a bit more background information on.
I've got quite a few questions actually to want clarification on. The reflector one is the one that's happening at Moorfield at the moment with the eye injections.
Yes.
You did say that they've moved on from the other two phases that they did with the injections in the eye, where they used the placebo in one eye, and in the other eye they used the actual gene therapy. You did say that they are now doing the same. They're not using the placebo in the other eye. They're using the injection in both eyes.
Yes.
Is that correct?
For the REFLECT study, it's a bilateral injection. So roughly around 2/3 of patients will get the injection of the active viral vector in both eyes. But in about 1/3 of patients, they will get injection in both eyes, but one eye will be the active viral vector, the other eye will be a vehicle, right? So overall, 3/4 of eyes will get the active viral vector, 1/4 of eyes will get the sham injection. And the reason why you still need a group of eyes that have not received the active agent is because you need to do the comparative analysis.
The second question, also pertaining to this course, is that what I don't understand is, I think it's been going on for about 3 years now, these injections. Have they not followed up with a second lot of injections? Why are they only just given one set of injections and not being followed up with another set of injections, with which you're putting in perhaps a bit more a gene?
Yes, I think essentially it goes back to the fundamental principle of how we think this is going to work. If you're giving the injection, and if what you feel is happening is that the replacement gene is being introduced into your nuclear genome, you're producing the protein gets into your mitochondria, then that should be sufficient to protect the cells within the optic nerve that were going to die, for example. So once you've rescued them, right, you should have been able to pull these cells back from the brink and prevent either the vision from getting worse or actually improve visual function, right? The difficulty with giving a second injection is not just theoretical, but if you give a second injection in one eye, there's a possibility that you've primed your immune system, and if you give a second injection, you might trigger quite a violent immunological reaction, right? I think this is unlikely, but if you're doing an experimental study where you still do not yet have the full answer about whether or not the drug is working, you have to tread very cautiously, right? And that's part where we are now. It's still not the end of this research program, not just from Gensight, but John Guy at Bascom Palmer is also running his own gene therapy program, and also Professor Li Bin in China who's doing the same. So I think there's a lot of groups working on this, and to me that's a good thing, because this means that there's accumulated evidence, and both in terms of safety and in terms of potential efficacy, both short-term and long-term.
My final question is that you did say from the outset that when this gene affects, it only affects, or usually only affects, the optic nerve. You use the word usually. Has there been cases where this has affected other parts, other nerves?
Oh, certainly. I think for Leber's optic neuropathy, it's quite clear that the optic nerve gets the biggest hit. But it is very well established that there will be a small group of patients who carry these mitochondrial DNA mutations that can develop other features that go beyond the eye. The most well-described association is between Leber's optic neuropathy and multiple sclerosis, which was first described by Professor Anita Harding from Queen Square, and it's sometimes called Harding's disease. So it's a very intriguing process where not only do you get the visual loss from the optic nerve involvement, but you also get changes within your brain, which is, if you only show the MRI scan, to a neuroradiologist, the radiologist will say this is MS, right? And, this is again a very complex topic, which I think we've touched upon this at a previous LHON patient. I'm pretty sure I covered this topic before. But here the difficulty is, how we manage the condition. Do we manage it as being MS, or do we manage it as being a rare mitochondrial problem? And this is where it's challenging having a rare disease. It's even more challenging having a rare subtype of a rare disease, right? But again, research is being done on this. But absolutely, fortunately, this is a smaller subgroup, but you do and you can get other extra ocular features.
So I actually had treatment for MS. And it definitely improved my sight. Well, it improved the MS.
Yes. Well, it is. I also have people that I've followed, and there are actually people in the room where clearly the thinking process is, if you at least, it can be controversial, but in my own thought is that if someone has this so-called LHON-MS overlap syndrome, they should be treated as having MS and therefore get all the disease-modifying drugs that would be appropriate for the stage and the severity of the disease.
Are they always extra symptoms? So if somebody presented with symptoms of MS but didn't have vision issues, could that be sort of LHON related or not? I
think not in my personal experience, because if you have someone with MS, it is possible that you can have MS without any visual problem, but it's very easy to rule that out, because if you do genetic testing and you don't have a LHON causing mitochondrial DNA mutation, then this is not LHON MS. And actually, after this was reported, people have looked into their patient cohorts where they've screened 100 patients with MS to see whether or not they had some of these LHON mutations, and the message from these studies is that this is uncommon. So the vast majority of patients with MS do not carry a LHON causing mitochondrial DNA mutation, but you can get the combination of someone with LHON and MS driven by this mitochondrial DNA mutation.
But equally, you could have a comorbidity where people have these as two entirely separate diseases.
Yeah.
And that's the difference.
And that's exactly, I think the reason why I gave the example of LHON MS is because in that specific association, there's very strong evidence. But there's also been studies out there saying, well, if you have LHON, or you have an increased risk of having migraine, or an increased risk of getting other possible complications, there it's much more difficult to be sure, because migraine is very common in the population. 30% of the population have migraine. So is your LHON mutation promoting that or not? So we need to be very careful whether or not whether someone with 2 or 3 different comorbidities is actually causally related to the mutation, or whether it's just coincidental, right? So someone could have diabetes and could have LHON, just because diabetes is quite common in the population. LHON is not that common, but you could have the two of them combined together. But based on the statistical analysis, the association of LHON-MS is, I think the association is pretty strong in terms of that there is a link there.
I had a question. A lot of these trials are based on the more common 11,778 gene. Mm-hmm. If they were successful, how well would it then translate to the other strands? For instance, if the Gensight was proved workable, could it then be used on the other things, or is the likelihood that that same thing would then not work on the other strands?
It's a very good question. The question is whether or not, if the gene therapy paradigm for 11,778 works, how easily applicable will it be to the other 2 main mutations, which is 3460 and 14484? The answer is, if the results are promising and show categorically that the treatment is working, then in theory you could use the same vector strategy to insert the other replacement gene. So instead of replacing ND4, which is 11,778, you could replace ND1 for 3460 and ND6 for 14484. So on paper, it will be very straightforward. And actually, as I mentioned already, there are other groups. There's the group of Professor Lee Bean that I know of, who's working on this. There's also the group of John Guy at Bascom Palmer, who is actually trying under a very interesting paradigm where instead of giving just the one vector with the one replacement gene, you have more than one replacement genes in the same vector, right? But remember, this is the academic side of the program. If you come up today with your new vector, say for ND1 3460, you will have to go through the same process again, of course, because safety is the most important thing, of checking whether it works in your model, doing all your safety study before you move on to the clinical trial. So from now to the next phase of whether a new vector is working for 3460, we have 10 more years ahead of us.
Hi Patrick. If the reverse gene therapy study was proven to be successful, would they be able to open that out to people that have been affected for more than 12 months without doing another trial for safety and efficacy?
It's a good point. I don't know the answer for this, and I think this will probably depend on the strategy of the company and how they approach their dossier when it comes to the European Medicines Agency. Because obviously you invest time and effort and lots of money into developing these programs, because at the end you hope you will make a profit out of it. As to what label the company is going to go for, I'm not sure, right? But we'll just have to wait and see. It's a very good point, actually, and when I see them in June, I will try and probe that question and let you know.
Thank you.
Hello, just wondered if you've had much experience of anybody having side effects of the idebenone. Our son had to get taken off it for a drop in white cell count, and his neutrophils in particular were quite low, so they had to take him off that. But I don't know how typical he is anyway, because he's a bit of he has extra symptoms too, so he has like dystonia in his hands and things, so he may just be not following much of a pattern.
Well, certainly I think any drug can have idiosyncratic reactions. The body evidence from the RODOS study or the retrospective series published out there and the more recent EAP and LHON study which is ongoing is that generally speaking this is a very safe drug that can be given for a long duration of time. But there's always been some worry regarding whether or not that in a few patients you can have deranged liver function, for example, it can affect your lipids, or sometimes it can be a drop in your white cell count. I've also had one of my colleagues who's reported that for one of his or her patient. So I think this can happen. It's sometimes a bit difficult to establish causality because there could be real reasons for that, but certainly I feel that it would be safe that if you're taking idebenone for a long period of time, or even if you're considering taking it indefinitely, that you should probably have a blood test once every year. Just make sure that your full blood count, that your liver function, that your cholesterol, everything is as it should be. And it's probably also good practice after a certain age, really, right? So I think it's rare, but kind of based on what I've heard, based on the fact that I know that some of my patients also had to be withdrawn from these studies because of these abnormal blood results, which are above the normal range, but probably it's not causing any damage, but we need to be on the safe side when it comes to these studies. I think it's worthwhile to have an annual blood check if you're taking this long term, right? This is my personal opinion. The company might have a different perspective and other clinicians might have different perspective. All right, thank you very much. All right.